in my laboratory working on pain. So we're asking a lot of questions about chronic pain, acute pain, the mechanism of pain, and we're trying to develop new treatments that will selectively and efficiently block pain. So the first question that one should ask is why pain? And according to latest definition of chronic pain is a disease of nervous system like Alzheimer, Parkinson, and many other diseases. In the United States and in Europe, about 30% suffers from chronic pain. So we have a disease with huge amount of people that suffer from, and we don't have efficient treatment. So in my lab, we're trying to understand the mechanisms or the plastic changes that nervous system undergoes as a result of chronic pain. We're using a variety of physiological techniques. We're using a recording from single cell or from group of cells in live animals during normal and abnormal pain stimuli in order to understand what kind of changes in cell connectivity chronic pain produces and how these changes are preserved to perpetuate and create the phenomenon of chronic pain. So in my lab I have 6 or 7 students and one postdoctoral fellow, and we are trying basically to target pain in all sites of the detection, transmission, and analysis. We're trying to understand how peripheral nerve ending detect noxious stimuli, again, in normal and pathological condition. In order to do so, we're developing a new, very exciting, high-end technologies to be able to record from very tiny nerve ends that are the size of half a micron. We're using cameras that initially was developed by NASA in order to record signals from very far away stars, and using the same chip in the camera in order to record activity of these terminals. We then follow propagation of these electrical signals on the pain neurons towards spinal cords. There we try to understand what's the nature of transmission of the information between peripheral neurons and central neurons. Again, normally and in pathological conditions. It's very well known that in chronic pain we have a very interesting plastic changes that basically enhance the connectivity between cells, both between peripheral neuron cells and within the local networks of central cells. And this is what we're trying to understand.